So far, all of our discussion of evolution has been based at the population level. Population level evolution is called microevolution because it's focusing on a small number of organisms or a relatively small proportion of the entire species. But if we go higher and we look at species level changes, and if we look at the production of new species and the long-term evolution of those species, we move into macroevolution. So in macroevolution, we start to look at how species form. So speciation is the process by which one species splits into two. In other words, a group becomes fragmented from the other and is no longer the same. Now, speciation is the process usually propelled by natural selection that produces the tremendous diversity of life on Earth. So if we're going to talk about speciation and how it occurs, we need to define what we mean by species. And we're going to do that using the biological species concept. In the biological species concept, we define a species as a group of populations who can interbreed in nature or potentially interbreed in nature, because sometimes we have populations that may not meet all the time. So a group of population whose members can interbreed in nature and produce viable, fertile offspring. So two groups have to be able to reproduce in nature, so naturally, and produce organisms that are going to live and also produce their own offspring. In other words, we might define a species as a group that is reproductively compatible or has reproductive compatibility. Now, there are a variety of other ways that we can define species, and our book does talk about a couple of them. So there are 7 or 8 other species concepts, and they're used for different situations. So for example, we might use the morphological species concept to assign species based on physical characteristics. Because biological species concept requires that we know something about interbreeding and something about the offspring. And there may be situations where we can't know that information. So for example, if we're looking at fossils or you only have one specimen, you might use morphological species concept and separate a species based on physical characteristics. We can use some other species concepts, like the evolutionary species concept is based on common ancestors and fossil record and whatnot. Phylogenetic species concept is based on DNA. So there's different reasons that we would use them. We're going to use biological species concept for our learning for this chapter. So the biological species concept is based on reproductive compatibility versus reproductive isolation. So new species form when reproductive isolation occurs. In other words, there are biological barriers that inhibit interbreeding. So those reproductive barriers are going to block gene flow, which in turn isolates those those groups, and allows each one along its own evolutionary pathway towards becoming so different that they are no longer reproductively compatible. So for example, these are the eastern and western meadowlarks, and they are considered separate species because they don't interbreed and produce viable offspring. Now, the reason that they don't interbreed is that they have different songs, and so they don't recognize one another as being the same species, and so they're not interested in something that's not the same. So there are different mechanisms by which these reproductive barriers can occur. They are pre-zygotic barriers and post-zygotic barriers. Remember from our genetics chapter that zygote is just a fertilized egg. So in other words, these are barriers that inhibit any fertilized egg from being made, or they are barriers that, even though a fertilized egg has been produced, still don't produce viable offspring. So these barriers to reproduction can lead to speciation because they isolate populations from one another. So we're going to talk a little bit about both the prezygotic and the postzygotic barriers. I would strongly encourage you to go back to your textbook in chapter 22 and read a little bit more about these, because there are a lot of great examples that I simply don't have time to go through in too much detail. So a prezygotic barrier is a barrier that's going to block fertilization. So usually this means that no physical mating is even going to happen. If a physical mating happens, then no fertilized gamete is going to result. So some of these are essentially basically choices kind of that an organism is making, where they're choosing their habitat, what types of behaviors they're having. So in habitat isolation, this might be that even if the organisms could potentially interbreed, they're choosing different habitats or different locations within their environment that keeps them separated. So for example, this fly, A and B, make their habitat on different fruits. And because those fruits are in season at different times, they are utilizing them at different times, they have different time frames for their life cycle, so they're simply not mating. You can also have temporal isolation where the mating seasons don't line up. So if the mating season doesn't line up, one is not going to be ready to mate when the other is, and so they can't mate with one another. You can also have behavioral isolation. So the western meadowlarks in the previous slide were behavioral isolation. They were performing a song as part of their mating ritual to attract a mate. Other organisms like the blue-footed booby example here does a particular dance to attract a mate. So again, if you're not seeing an individual perform the correct mating ritual or not hearing the correct song, you're not going to recognize them as the same, and you're not going to interbreed with them. You can also have a mechanical isolation. So what this means is that mating simply can't happen due to physical characteristics. So for example, these snails, I love this example, have different directional curls on their shell, which position their sexual organs on opposite sides. And so there's no direction that they can mate where this is functional. And that was due to a single gene mutation. One of them acquired a mutation that caused the shell swirl to turn the other direction. And boom, new species. And then we've got gametic isolation. So basically this means that the sperm and the egg are completely incompatible. And so the sperm cannot successfully fertilize the egg even if mating takes place. We can also have post-zygotic barriers to reproduction leading to speciation. So this means that physical mating can actually happen, and an organism may actually, or the egg is actually fertilized, and an organism might actually be born. So post-zygotic barriers happen when there's no viable or fertile offspring. So sometimes that means that the offspring doesn't survive to reproduce or doesn't survive ever. But it also might mean that an organism survives but then is sterile itself. So there's a couple of different varieties of post-zygotic barriers. One is reduced hybrid viability. So the hybrid is the offspring of the two groups. Reduced viability means that they're not going to survive long enough to reproduce. Reduced hybrid fertility means that any organism that is actually produced cannot have its own offspring. So our example here is a donkey and a horse producing a mule. Mules are perfectly viable on their own, perfectly functional, but they cannot produce, they can't mate with other mules or horses or donkeys to produce viable offspring. And then you can also have something called hybrid breakdown, which is where basically the offspring are incompatible with anybody else. And so the only organisms they can mate with are themselves. But usually, if that happens, they're not. They're still not viable. They're not a population that is going to keep going. And of course, if none of these conditions are met, then you are not an isolated population and you're not separate species. So if you do produce viable fertile offspring, then the process of speciation has not finished. So as these barriers, prezygotic and postzygotic barriers, are affecting populations, we're going to end up with new species. Now, based on these prezygotic and postzygotic barriers, there are actually two ways that new species come about. And these are allopatric and sympatric speciation. In allopatric speciation is essentially a prezygotic barrier in which gene flow is inhibited by some sort of geography, some geographical feature that is physically separating organisms so that there is no possible way for them to interact. So in allopatric speciation, a population forms because it becomes separated and completely isolated from another such population, and then each one is free to diverge along its own evolutionary route. And as they are diverging, different mutations, different adaptations, different natural selection, different genetic drift is going to happen to both of those populations. So a great example of this in your textbook is of two squirrel populations that are separated by the Grand Canyon. So the Grand Canyon is too large for the squirrels to cross, and so they don't interact, they don't mate. And this has led to two distinct squirrel populations on either side of the Grand Canyon. In sympatric speciation, populations are not geographically separated from one another and can still encounter one another. So in sympatric speciation occurs without that geographical separation. Now, it's much less common because if they can encounter one another in the early stages, they're probably exchanging genetic material, which means they're probably not diverging very quickly. This doesn't happen as often in animals, although there are some great examples of this in animals. But it does usually, when it does occur, it usually occurs in plants. And there's a lot of weird genetic mechanisms that happen in plants. Plants can survive with duplicated genomes, for example, or parts of genomes. And so this can actually happen in plants much, much easier and much, much faster in animals. But our example here for sympatric speciation is two fish populations that, although they seem to be generally divided by that ridge in the middle of their pond, they can still cross, which means that they can interact, they can pass genes back and forth. But if sympatric speciation is happening, it's usually based on one of the prezygotic barriers that we were talking about, some sort of habitat differentiation or behavioral isolation that causes them to not choose to mate with one another, even though they do come in contact with one another. So how fast does speciation happen? Well, in some cases, it can be marked by one specific gene that makes them reproductively incompatible. like the example of the snail shells that I gave you. In plants, sometimes genetic changes happen in one generation. So speciation can happen very quickly. Speciation, depending on whether it's allopatric or sympatric and what genes are involved, might take years and years and years. It really kind of depends on the selection pressures, the differences in those populations, the differences in the environment. And so there is no real definition for how quickly speciation happens. It happens on a case-by-case basis, sometimes quickly, sometimes not.